What is up, y'all? Welcome to another PowerPoint Lab Animations. Today we are talking about PT and PTT, but this video is going to be a little bit different because we're going to focus on the history of coagulation theory more so than modern laboratory testing. I am a big proponent of understanding where we've been to better understand where we are today, and I think historical context like this can make some of these concepts a little stickier. So ideally, you'll come away from this with a better handle on the modern PT and PTT tests and some fun trivia you can share with your lab friends. As always, what you're going to see today was created and recorded in PowerPoint by a real person. I do have one small caveat today, and it's that I asked ChatGPT to make some clot clip art, which you can see on the screen now. If you work the coag bench in a modern hospital lab, you probably run dozens, if not hundreds, of PTs and PTTs every day. What you may not realize is that both of these tests are modifications of the prothrombin time test created in 1935 by Armand Quick. These tests are almost 100 years old, and they barely changed since that original publication. At the time, Quick was trying to design a diagnostic test for hemophilia, and if we're going by that stated goal, he failed. But by almost any other metric, he crushed it. We still use his prothrombin time today, and what he did find catalyzed the invention of the PTT, which does actually diagnose hemophilia. So how did an essentially failed experiment produce modern coagulation testing? I'm going to answer that, but we have a few detours along the way, so let's get into it. The thing that initially sent me down this rabbit hole is this infographic that you're seeing now, which is courtesy of Stago. If you're watching this video, you probably know Stago is one of the big dogs in the coagulation analyzer field. Stago is actually pretty big on educating their end users. They even made a board game called Clot in the 70s, where you make your way around a board game by answering questions about the coagulation cascade, with the winner being the first one to form a fibrin clot. It's not a fun game, but it is very educational, and I wasn't surprised to find something like this on the Stago website. What was surprising was what I found when I did some very basic Googling, trying to get more info on the second item on this timeline, which is characterized as the dawn of coagulation research. Now, I do not want this video to be a point-by-point takedown of this infographic, and that might be coming up at a later date, but there are some issues with giving Andrew Buchanan credit for discovering tissue factor and simultaneously creating the field of coagulation research. One of the first things that caught my attention when I started looking into this was this 1998 article by James Markham in the still in-print journal, History and Philosophy of the Life Sciences. In this article, Markham is going to contend that credit for the discovery of tissue factor is misplaced and should actually go to another guy named Alexander Schmidt. And he makes some pretty compelling points that we're going to talk about here. Buchanan is by no means the first person to study coagulation, as this infographic would have you believe. The mystery of blood clotting has been the subject of scientific theory and debate for just about as long as we've had scientific theory and debate. That blood does not remain liquid when outside the body is an immediately observable phenomenon. What's more, this phenomenon seems to happen spontaneously, and early coagulation researchers called the spontaneously formed substance fibrin. And spontaneity is an important piece of this because, as it turns out, many early coagulation researchers were also invested in the historical debate over spontaneous generation. If you're familiar with the historical theory of spontaneous generation, you probably know the story of Spallanzani and his rotting meat, or Louis Pasteur and his rotting wheat. Spallanzani and Pasteur were trying to disprove the idea that life was continually and spontaneously being generated from non-life. Coag researchers in the 19th century aren't necessarily using fibrin formation to disprove spontaneous generation. There is a lot of overlap here, since coagulation is a seemingly spontaneous process, and by characterizing this process, researchers were in some small way characterizing the nature of life and existence as we know it. This connection actually helps to explain why reading your coagulation chapter sometimes feels like reading the Silmarillion. 19th century coag researchers were trying to characterize enzymes before the concept of enzymes had been birthed. Pasteur used the word ferment to describe the vital force in the living yeast cells that created alcohol. The term ferment also applied to the action of amylase on germinating grain and to the action of rennet in the coagulation of milk into cheese. The use of ferment in this way was eventually replaced by the word enzyme, which borrowed from the Greek word zymos, meaning leavened by yeast. Side note, this is also why the study of fermentation and the official publication of the American Homebrewers Association are called zymergy. So it was a real saying it with your chest moment when, in 1872, Alexander Schmidt isolated a catalytic compound and called it Das Fibrin ferment. Alexander Schmidt was working under working under none other than Rudolf freaking Virchow when he characterized what he called the fibrin ferment. We don't have time to get into Virchow here, but he is on the Mount Rushmore of pathology. At the time Schmidt published his initial paper, Virchow's lab was focused on thrombosis. Virchow had previously proposed that insoluble fibrin was formed from a soluble fibrinogenous substance upon the exposure of blood to oxygenated air, so Schmidt was working on defining and characterizing that process. Schmidt ends up discovering that the fibrin clot formation is dependent on three factors: fibrinogen, the soluble precursor to insoluble fibrin; fibrinoplastin, now known as tissue factor; and what he called the fibrin ferment, now known as thrombin. Schmidt was eventually able to show that the rate of fibrin formation was directly influenced by the amount of fibrin ferment, and that the ferment was not consumed by the reaction. It is Schmidt's work on the fibrin ferment that led to our first unified theory of coagulation, and he was recognized by his contemporaries as an important name in coagulation. He ended up devoting his career to coagulation and oversaw a lab that continued to study coagulation after his landmark paper. So how and why does Schmidt get left off of this infographic? Well, it turns out that Schmidt had possibly unknowingly, but probably knowingly, duplicated the work of the previously mentioned Andrew Buchanan. About 30 years before Schmidt, Buchanan had discovered that he could create a fibrin clot by mixing serum and a washed blood clot. For Buchanan, this is more of an incidental finding. The focus of his research was tissue growth and nutrition. In fact, he did not really pursue this finding further in any future research. When Buchanan made this observation, he proposed that a factor from serum combined with a factor from the tissue, and the result was a fibrin clot. However, he did not isolate either factor and did not characterize the reaction as catalytic. A comparison was made to rennet, but that was made by an editor of the journal in the remarks section following Buchanan's research. So Buchanan's work was significant as far as the historical record is concerned. He is the first person to make a fibrin clot using only stuff that was present in human fluids. This was a big step forward, even if it didn't have an immediate impact on coagulation research. Schmidt actually acknowledges Buchanan's work in his initial publications, and it is probably more accurate to think of Schmidt as extending Buchanan's work as opposed to Schmidt arriving at his experiment independently. Schmidt started with Buchanan's observations. Mixing serum and fluid from a washed clot produced insoluble fibrin, but Schmidt was able to isolate the so-called fibrin ferment and even demonstrated its catalytic activity. Ultimately, the story of how Schmidt gets replaced in the historical record is kind of uninteresting. This English physician and researcher named Gamgee made a point of retroactively giving credit to his countryman Buchanan for the discovery of the fibrin ferment, and he was successful, as evidenced by the fact that this Stago graphic doesn't even mention Schmidt. So Stago, and I know you're watching, I'm proposing an update to this infographic. We don't have to kick Buchanan off. He still gets credit for something, but we can't leave out Schmidt. My new entry would read, in 1872, extending the earlier work of Scottish physician Andrew Buchanan, Alexander Schmidt demonstrated the catalytic nature of the so-called fibrin ferment. Schmidt's work confirmed Buchanan's observation and inspired the first theory of coagulation as an enzymatic process. All right, so we've corrected the historical record as far as Alexander Schmidt is concerned, and because this is not a video about the Stago infographic, I'm moving on from that. Both Schmidt's and Buchanan's work were foundational to the first unified theory of coagulation as proposed by Paul Morowitz. This is Morowitz's claim to fame, and his theory was pretty prescient for somebody working in 1905 when the concept of an enzyme was still in its infancy. Morowitz proposed that blood coagulation was a two-step process. The first step was the formation of thrombin, which was Schmidt's fibrin ferment. He proposed two methods for thrombin formation from prothrombin. One involved Buchanan's washed clot fluid, which Morowitz called thrombokinase, and we now call tissue factor. This is another consequence of coagulation theory outpacing enzyme theory. At the time, kinase was a general term for a substance that promoted a chemical reaction, not the specific class of enzymes it refers to today. To make things even more confusing, some researchers preferred the term thromboplastin. So when you're reading these historical papers, tissue factor, thrombokinase, and thromboplastin are often referring to the same thing. Morowitz proposed that this thrombokinase could convert prothrombin to thrombin in the presence of calcium. He also proposed that thrombin could be formed from prothrombin by the action of thrombogen, which would later be identified as factor 10. In other words, he proposed two pathways to thrombin formation, one involving a factor outside of or extrinsic to the circulating blood, and one inside of or intrinsic to the circulating blood. And if I've done my job to this point, some light bulbs should be going off right about now. The second step was the formation of fibrin from fibrinogen under the action of thrombin in the presence of calcium. If you've studied coagulation in your program already, you should recognize this as the extrinsic, intrinsic, and common pathways that we would now recognize today. So props to him. He was cooking with that one. Moritz's theory almost immediately gains traction, and in 1935, Armand Quick was using Moritz's recipe to try to find a test that could diagnose hemophilia. By 1935, thrombokinase or tissue factor had been isolated, and researchers could use it as a reagent in their experiments. At this time, bleeding disorders like hemophilia were diagnosed by seeing how long it took blood to clot after adding calcium. This worked, but it was time-consuming and subjective. Quick had observed that out of Moritz's 4 proposed factors, calcium and fibrinogen were almost never the cause of bleeding in hemophiliacs, and that tissue factor had no thrombogenic properties on its own, meaning you could add as much tissue factor as you want to fibrinogen, and you're never going to get fibrin. Based on these observations, Quick sought to devise a way to test prothrombin levels in hopes that the results could more easily discriminate between hemophiliacs and normal patients. And again, he did not end up doing this. Instead, he completely corrected the abnormal clot times of hemophiliacs. He basically showed that if you added enough tissue factor to calcified blood, the clot time could be reduced to 22 seconds for all patients, including hemophiliacs. By correcting their clot times, Quick had demonstrated that hemophiliacs have normal prothrombin, which meant that there was yet another factor that needed to be discovered to explain hemophiliacs' abnormal bleeding. Despite not being a diagnostic test for hemophilia, Quick's prothrombin time replaced the clot time as a baseline screening test for his basic method is still in use today. Quick added calcium and tissue factor to citrated whole blood and measured the time to clot formation. Today, we perform the prothrombin time, or PT test, on plasma, but the other components are the same. Quick's finding that hemophilic clot times can be corrected with the addition of tissue factor was pretty revolutionary. Ruling out thrombin as the problem in hemophilia opened a whole new line of research. Pretty soon after Quick published his prothrombin time, a group of researchers working out of UNC Chapel Hill and led by Kenneth Brinkhouse started working on modifications that could diagnose hemophilia. The first step was a test called the 2-stage prothrombin utilization rate, and this is going to be where we live up to the name of the channel today. So we have to get caught up on some of the basic assumptions and observations Brinkhouse and his team were working under when they came up with this test. The word cascade had not entered the lexicon to this point, but they knew this was a stepwise process that involved enzymatic reactions. The ultimate substrate of this stepwise reaction is fibrinogen, and in a vacuum, the formation of fibrinogen would be the rate-limiting step. You can't make fibrin without fibrinogen. They knew then, as we know now, that there is a way more fibrinogen in human blood than is needed for normal bleeding. In plasma, it's right up there with albumin as far as concentration is concerned. They had also figured out that there was way more prothrombin present in the blood than was needed to make the necessary amount of thrombin to stop bleeding. They knew this because you could recover prothrombin from the serum of clotted blood. Brinkhouse and his team proposed that if you could measure the excess prothrombin, you could discriminate between hemophiliacs and normal patients based on the residual prothrombin remaining after blood had clotted. This was the basis of the prothrombin utilization rate test, which was run in 2 phases and was the immediate precursor to the modern PTT. The first stage on this test was a prothrombin time. This produced the serum that would be tested in the second stage. In the second stage, they ran a series of prothrombin times with the addition of fibrinogen on the recovered serum, and what they found was that hemophiliacs clotted much more quickly in the second stage than the normal patient. Brinkhouse and his team proposed that normal patients utilized more prothrombin in the first stage because of the elusive anti-hemophiliac factor, which they would eventually get credit for discovering and was eventually renamed to factor 8. I think this is a really cool finding because it illustrates how hemophiliacs have this disconnect between the extrinsic and intrinsic pathway, and that is really the root of their bleeding issues. They didn't know this at the time, but Quick's prothrombin time was able to correct hemophiliac patients' clotting times because the amount of tissue factor being added was overwhelming our normal systems of control. In the normal bleeding response, small amounts of tissue factor are introduced into the bloodstream, and the result is lots of factor 10 and thrombin being generated. As Quick demonstrated, tissue factor is almost a little too powerful, and normally we shut down the extrinsic pathway pretty quickly through this protein called tissue factor pathway inhibitor. TFPI pairs up with this excess activated factor 10, and this complex shuts down the tissue factor and factor 7 complex that we call the extrinsic kinase. At this point, in normal patients, the intrinsic pathway takes over so that we can fully stabilize the fibrin clot. Hemophiliacs don't have that second stage. They have no factor 8 to take over for the extrinsic pathway that's been shut down by TFPI. This observation also kind of illustrates why massive trauma can lead to DIC. If you have massive trauma, then you can end up with massive amounts of tissue factor being introduced into circulation. So much so that it can overwhelm our normal mechanisms of control. Just like almost all the prothrombin in a normal patient sample is consumed in the prothrombin time test, all the coagulation factors will be consumed in the patient if too much tissue factor is introduced into the bloodstream. Brinkhouse and his team unknowingly observed this disconnect when they found excess prothrombin in hemophiliac serum, and from there they arrived at the modern PTT. By 1953, Brinkhouse and his team were pretty sure that anti-hemophiliac factor was real and that its absence was responsible for the excess prothrombin that could be found in the serum of hemophiliac patients. They had also been experimenting with different formulations of tissue factor. The thing that makes tissue factor is a phospholipid called cephalothin. As you can probably guess, this is normally present in cell membranes, especially those of platelets. It's also present in different amounts in different types of organs, and it's still true today that no formulation of tissue factor is created equally. Eventually, they found that if you diluted the tissue factor enough, or only included the phospholipid part, then you could almost eliminate its influence on the prothrombin time. Brinkhouse and his team used what they call the partial thromboplastin as a modification of Quick's prothrombin time to diagnose hemophilia in a one-stage assay that they called partial thromboplastin time, and we now call the PTT or APTT. This partial thromboplastin simulated physiological coagulation much more closely. Small amounts of thromboplastin were added to patient plasma, just enough to start the process, but not enough to overwhelm the system. Patients with an intact intrinsic pathway have a normal PTT, and patients missing factor 8 or 9 have a prolonged PTT. And that's my version of the history of coagulation testing. Almost 100 years later, these tests have barely changed, which is a fairly unique thing in the lab. Today, we think of the PT as being used to screen for deficiencies in factor 7 and to monitor for patients on warfarin. The PTT is still used to screen for hemophilia and is used to monitor patients on heparin. Got some links to sources in the description. Most of the images of all the white guys are from Wikipedia, and I did borrow a couple of diagrams from Pearson. Let us know below if there are any other lab tests or lab-related concepts you'd like to see animated in PowerPoint. Thanks.